Everyone, welcome to my channel Angels Nursing Decodes. In this video, we are going to discuss about food poisoning and food intoxication. Epidemiological features or clinical characteristics of food poisoning. The first one is history of ingestion of common food. For example, in family function, hostel, hotels, etc. And the second one is a group of persons being affected simultaneously. Next, similarity of signs and symptoms in the majority of cases. Common symptoms are vomiting, diarrhea, pain in the abdomen, fever, etc. Short incubation period. Absence of secondary cases. Laboratory tests are rarely required. Next, we can see the various types of food poisoning. First one is bacterial food poisoning. So, this is caused by taking contaminated food, may be infective or toxic. Infective means here the organism enters inside the body through the food, and there it will produce toxin, and then it will leads to pathological reaction and finally results in clinical manifestation. For example, Salmonella, Clostridium perfringens, Vibrio parahemolyticus. So here, in infective cases, the organism enter inside the body through food. Then it will produce toxin inside the GI tract, and there may be disturbance in the GI tract. Irritation will occur, and due to that, the symptoms will appear. And the second is toxic, due to already formed toxin in the food. Okay, like if the temperature is not maintained properly in the refrigerator, or if it is not stored properly, some toxic substance are produced in the food. So there, Staphylococcus aureus, Clostridium botulinum, Bacillus cereus. Okay, these organism will produce those toxins, and if we are taking those food, then it will leads to food poisoning. Next is non-bacterial food poisoning. It may be due to chemical poisoning. For example, pesticide, arsenic, mercury, etc. Next, third type is due to certain plants and seafoods. We have discussed already in mushrooms. There are two types, edible and non-edible. If we are taking the non-edible form, then it will lead to food poisoning. And the solanine, it is otherwise termed as green potatoes. This is also will irritate the GI tract. Next, we can see the common bacterial food poisoning and their source or common foods and clinical features. The first one is Salmonella food poisoning. So here, if a man is infected with this Salmonella typhi and that person is preparing the food, then what happen? It will be spread to other persons. So the food source which carries this microorganism is meat, milk and milk products, sausages, custards, egg and egg products. So the food is contaminated with urine of rats, and mice is also responsible. So sometimes if the rats are common in our home, means they will be passing urine. So in that also the microorganism will be present. Okay, due to that it will spread from one person to another. Then the clinical features of this Salmonella food poisoning were sudden onset with chills, fever, nausea, vomiting and profuse watery diarrhea. Sometimes there will be blood also in stools. Next is staphylococcal food poisoning. Here the persons, they will be having boils or carbuncles, or the animals, they will be having this boils or carbuncles, and if that fluid is in contact with food, or if those persons are preparing the food, then it will be spread to others. The food involved were salad, custard, milk and milk products. In this staphylococcal food poisoning, the symptoms were sudden onset of vomiting, severe and violent it will be. Okay, vomiting means it will uncontrolled. Suddenly it will occur as well as it will be uncontrolled. Next, abdominal cramps will be there, diarrhea, but rarely fever. Fever is very less common in this type of food poisoning. Next is botulism. So here soil and dust and the intestinal tract of animals. Here the organism will be present in the soil, dust or intestinal tract of animals. So the food which is responsible for this transmission is home canned vegetables, smoked or pickled fish, homemade cheese. So it is mainly present in homemade dishes. Okay, so if we are preparing the food in the home and packing for long time, here the organism will be developed. The symptoms were dysphagia, diplopia, ptosis, dysarthria, blurred vision, muscle weakness, quadriplegia, and here the GI symptoms very slight as well as the no evidence for fever. Next is Clostridium perfringens food poisoning. Dust and soil is the reservoir of spores, and here if we are recooking the food again and again, for example meat or like poultry, if we are using the food and again if we are heating means it will become food poisoning. Next, the symptoms were moderate diarrhea, sometimes abdominal pain will be there, nausea, vomiting, fever will be rarely present. Next, Bacillus cereus food poisoning. It is present in soil and food grains, mainly cereals, raw, dried and processed. Okay, so here if it is present in grains, so it will be survive cooking and germinate and multiply in favorable temperature. Okay, so the symptoms were two form. One is emetic and the other is endric. In emetic form, there will be signs of vomiting, and in endric form, there may be presence of fever. Next is food intoxication features and how we can prevent it and how we can control. Okay, let's see that. So the food intoxication occurs in two categories. Either it may be due to naturally occurring toxins in the food grains or due to toxins produced by the fungi in the food grains. So if it is naturally occurring toxin means they were lathrism, epidemic dropsy, endemic ascites, toxic polyphenol. And if it is produced by the fungi in the grains means they were aflatoxicosis, ergotism. So first we can see the food intoxication, lathrism. Okay, so the toxins and food involved characteristic features were this. If we are taking the kesari dal, okay, so it's a consumption of pulse, lathyrus sativus, that is means kesari dal. So it contains toxin beta oxalate aminoalide, BOAA, beta oxalate aminoalide. So it will be affects the nervous system. Okay, so it will leads to paralysis. And how we can prevent this means we can soak this grains or pulses in the hot water. Okay, so before consumption we can soak this in hot water. It is known as steeping method, and parboiling, vitamin C prophylaxis. So what happened? This toxin levels will be removed and it will be helps the pulse in free from toxins. Okay, so this lathrism, it is present in kesari dal and it contains the toxin beta oxalyl aminoaline. It will leads to paralysis of legs. And how we can prevent means we can remove the toxin by soaking the pulse in hot water. The method is known as steeping method and parboiling vitamin C prophylaxis. Next, we can select cultivation of pulse strains with low level of toxins. Okay, these were the two methods. One is removal of toxin by soaking the pulse in hot water, that is known as steeping method, and parboiling vitamin C prophylaxis. And the second method is banning the crop which contains more toxic level, or we have to select the cultivation of pulse strains with low level of toxin. Okay. Next is epidemic drops. Here the mustard oil is contaminated with argemone oil. So it contains the toxic substance alkaloid sanguinarine, and the symptoms were swelling of the legs, then diarrhea will be present, dyspnea, cardiac failure and sometimes death. So how we can find this argemone oil means nitric acid test and paper chromatography. Okay, these two tests, nitric acid test and paper chromatography. These two tests helps for detecting argemone oil. And how we can prevent it means we can avoid the mustard oil. Okay, maximum we can try to avoid this, or we have to remove the argemone weeds while growing along with this mustard seeds. Okay, so and we have to pack the mustard oil and which is free from argemone, like that. In cooking oils, we have to mention that argemone free. Okay, so it contains the toxin. It is mustard oil is contaminated with argemone oil. It contains the toxic substance sanguinarine, and it will be leads to swelling of legs, diarrhea, dyspnea, cardiac failure, and death. Okay, the test used to detect this argemone oil is nitric acid test and paper chromatography. And how we can prevent means maximum we can avoid mustard oil for cooking, and we can remove the argemone weeds growing along with mustard seed crops, or we can pack the cooking oil with label argemone free. Next is endemic ascites. So the millet Panicum millei, locally known as Ganthe, contaminated with seeds of Crotalaria. It is commonly known as Junjunia. Okay, so it contains hepatotoxin named pyrrolizidine alkaloids. So the symptoms will be ascites and jaundice. So how we can control this means educate the people in the affect area by deweeding of Junjunia plants which grow along with the staple. Okay, so we have to remove the seeds of Junjunia so that it will be protect the millet Panicum millei. Okay. Next is alpha toxicosis. Here the fungi Aspergillus flavus parasiticus infest food grains such as groundnut, maize, parboiled rice, sorghum, wheat, rice, tapioca under conditions of improper storage and produce aflatoxin. It also characterized by hepatitis, cirrhosis of liver or enteritis. So how we can prevent means we have to store the food grains in proper dry containers, and the moisture content should be below 10%. and we should not consume the food if it is fungus contaminated, and we have to educate the people regarding health hazards. Next is ergotism. The ergot fungus claviceps fusiformis infest food grains such as bajra, rye, sorghum and wheat during flowering stage. So the symptoms were nausea, repeated vomiting, giddiness, drowsiness. So how we can control this means we can remove the ergot grains by floating in salt water, or we can hand pick. Okay. Next is public health response to foodborne diseases. So early detection, management and referral. We have to identify early and we have to go for treatment. Then follow up is there, means we have to advise for that. Next, if there is any cases is there, means we have to immediately report it and investigate about it. Then health educate the public regarding foodborne diseases. Next, we can see few questions in this topic. Which is not an epidemiological feature of food poisoning? Group of persons being affected simultaneously, similar signs and symptoms, secondary cases are common, short incubation period. Here the option A is epidemiological feature of food poisoning and B is also the epidemiological feature of food poisoning, and D is also short incubation period is also a feature of food poisoning. So the answer is secondary cases are common. Next is outbreak of epidemic dropsy may occur due to contamination of which food item and with what? Mustard oil contamination with argemone oil. Next question. Kesari dal lathyrus sativus is associated with which food intoxication? The correct answer is lathyrusm. Next question. Canned food is mostly associated with which bacterial food poisoning? Salmonella, staphylococcal, botulism, bacillus. The correct answer is botulism. Next question. Mention 4 common symptoms of foodborne infection. Answer is diarrhea, nausea, vomiting, abdominal cramps, fever. Next question. What are the levels in the food chain when food can be contaminated? The answer is food production and processing, food preparation and handling, improper storage of food. So during these stages, the food poisoning will be occur. So I hope you all enjoyed this lecture. Thanks for watching and keep supporting. Please subscribe to my channel. Thank you.